Hello friends, I am Shubhangi Rohatgi, PGT Biology, KV1 AFS Hindon, and I am here to welcome you all in the session 2 of chapter 6 class 11th biology, that is anatomy of flowering plants. In the session 1, we had studied about different types of plant tissues that are present in a plant. We began with the discussion on meristematic tissues. We discussed that meristematic tissues can be divided or classified into primary or secondary meristematic tissues depending upon their origin and position. When we classify meristems according to their position, we say that meristems can be apical or intercalary. Both these types of meristems are primary meristematic tissues. The lateral meristematic tissues belong to the kind that is the secondary meristematic tissue. The function of the meristem is to cut off new cells for the growth of the plant body. They continuously divide to produce new cells by mitosis, and these new cells later on elongate and mature, finally get differentiated by acquiring their proper structure and function. Then the other type of tissue that is present in the plants is the permanent tissue. Permanent tissue can be of simple or complex type. When the permanent tissue is made up of only one type of cells, it is said to be simple permanent tissue, while when it is made up of more than one type of cells, it is known as complex permanent tissue. Simple permanent tissue can be made up of living or dead cells. When all the cells are living in it, it can be either parenchyma or collenchyma. Parenchyma and collenchyma, both the tissues have living cells in them, but the difference lies in the thickness of the cell walls. While the cell walls of the parenchyma are quite thin, the cell walls of collenchyma have irregular thickness at the corners. The dead simple tissue is the sclerenchyma. It consists of sclerids and fibers, and the cell walls are so much so lignified that the lumen is empty and the cells become dead. Finally, talking about the complex permanent tissues as I just said are made up of more than one type of cells. So, under this heading we studied about xylem and phloem. Xylem has 4 xylem elements, that is tracheids, vessels, xylem parenchyma and xylem fibers. Phloem is also made up of 4 types of elements. They are sieve tube elements, companion cell, phloem parenchyma and phloem fibers. So, this was all that we studied in the last session. Also, I had given you some questions to think over. I hope you all have tried to find answers for the same. Let us discuss them in short. So, the first question was, does the classification of meristems on the basis of origin and position seem to overlap? If yes, then how? You were asked to decide whether it is good or correct to classify the meristems only on the basis of origin or only on the basis of position. You had studied a few names like primary meristem, secondary meristem, apical meristem, lateral meristem, so on. So, did not you find that these nomenclatures are overlapping? Of course, they are. Let us see how. When we classify the meristems on the basis of origin, we classify them as primary and secondary. Primary one is the one which is developed earlier, and the derivatives of primary meristem give rise to secondary meristem later in the life cycle of the plant. So, on the basis of origin, we have primary and secondary meristems. Now, these primary meristems, they cut off new cells to make the primary plant body, and from the derivatives of the cells of primary meristem, apical meristem and intermeristems are derived. So, on the basis of position, when we classify meristems, we divide them as apical, intercalary and lateral, but when we look as a whole in toto, we see that apical meristem and intercalary meristems are both parts of primary meristem, while the lateral meristem is a type of secondary meristem. So, in this way this classification definitely overlaps. The next question was, have you ever heard that meristem cultures are done in order to obtain virus free plants? What characteristics of meristematic cells do you think help them to stay virus free? Well, it has been found that the spread of virus in the plant tissues does not keep up with the fast meristematic growth. You know, the viruses need a route to spread in the plants. Either they spread through the phloem, or for cell to cell movement they use the plasmodesmata. Now, if they are moving through plasmodesmata in the meristems, they won't be able to cope up. Why? Because meristematic cells are dividing at such a fast rate, they are so metabolically active that the viral movement through the plasmodesmata from one cell to another cell is quite restricted. So, it seems to be very low to cope up with the continuously dividing meristematic cells. Other reasons are absence of vascular system. As I just said that viral cells or viral particles, they move through phloem, they take the route of phloem to spread to different parts of the plant. But in the meristematic tissues there is no phloem, there is no vascular system. So, they get a restriction here and fail to replicate there in the meristems. Also, the meristematic cells are very highly metabolically active. As a result, viral particles fail to replicate in these cells. Further, it has been hypothesized that if there is any virus inactivating system operating in the plant, if a certain part of plant defense mechanism is acting as virus inactivating system, then this system has higher activity in the meristematic tissues than any other region. This has been proposed. Obviously, it will pay the plant to keep the meristems virus free, because the new cells are being produced here and it would be good if they are virus free. Finally, it has been seen that auxins play an important role to keep the plant cultures virus free. So, a high endogenous auxin levels in shoot apices may inhibit viral multiplication in the meristems. So, these are the reasons for which we do not find viruses generally growing in the meristematic cells. The next question that was given was, find out what is totipotency, which type of cells should be most suitable for tissue culture and why. I hope you have heard this term totipotency earlier also. So, totipotency is the phenomena by virtue of which living cells of a plant possess the potential of producing an entire new plant. This is the basis of plant tissue culture. We take out some cells from the plant and culture them in a nutrient media. We give them good conditions, temperature, pH and other environmental factors are also regulated, and then we see that within a few days a mass of cells arises from those earlier cells which were used for tissue culture. This mass of cells is known as callus, and later on with further development we see that whole new plant can arise from this callus. So, totipotency is this phenomena by virtue of which a whole new plant can be produced from the living cells of the plant. Which cells should be most suitable for carrying out tissue culture? Of course, parenchyma, because we had discussed it earlier that it has ability to differentiate. We had discussed that if parenchyma are stimulated properly, they can be made to divide again. Then you were asked to differentiate between xylem and phloem. From the discussions in session 1, you must have been able to write down many differences. Let us discuss a few of them. Firstly, the movements in xylem is unidirectional from roots to aerial paths, but the movements in phloem are bidirectional. We all know that in a plant, a tree or herb or shrub, any plant, the roots absorb water and minerals, and through xylem it is taken to the other aerial parts of the plant. So, xylem movement is always unidirectional. On the other hand, talking about phloem, what does phloem do? Phloem carry the photosynthetes. Now, the photosynthetes are produced in the leaves, and generally we think that from leaves they are to be taken to the roots, that are the main storage organ or other storage organs. So, the movement is from source to sink. But this is the case when the photosynthesis is taking place normally and continuously. What when the season of fall comes? What when the leaves are fallen? It is autumn and there are no leaves to carry out photosynthesis. What will happen at that time? At that time, the storage organs like roots, etc., they will now act as the source, and now the photosynthetes will be carried out to all the parts of the plant. So, in this way, phloem transport becomes bidirectional. It will always be from source to sink, but as we have seen just now that the source and sink, they change according to the season, according to the needs of the plants. When the food is being produced, the source will be the leaves. When the stored food is being used, then at that time the source will be the storage organ from where the food is being taken to be distributed to other parts of the plant. Okay. Moving on, we see that in xylem the conducting elements are tracheids and vessels, and they are dead, while in phloem the conducting cells are sieve tube cells and they are living cells. Xylem has lignin in its wall, while no lignin is present in the phloem, and since the lignin is present in xylem, it provides mechanical strength. However, since no lignin is present in the phloem tissues, it does not serve to provide the mechanical strength. As I said just earlier, that conducting elements in xylem are two types, that is tracheids and vessels. On the other hand, here the conducting element is only of one type, that is the sieve tube. The next pair in which you had to differentiate was collenchyma and sclerenchyma. Well, we know that collenchyma tissue is made up of living cells, while sclerenchyma is generally composed of dead cells. Since they are living, the cells here have protoplasm, while sclerenchymatous cells are empty. They just have an empty lumen. Here in collenchyma, the wall thickening is not uniform, but in sclerenchyma, the wall thickening is uniform. The thickenings here are not made up of lignin, while the thickenings here are made up of lignin, and cellulose may also be present. Finally, the collenchyma has wide cell cavity, whereas in sclerenchyma the lumen or the cell cavity is comparatively narrower. Beside these, there are many other differences also, like collenchyma serves to provide mechanical strength as well as elasticity. On the other hand, sclerenchyma is only a mechanical tissue. It is so stiff that it cannot provide any elasticity, it only provides rigidity. However, collenchyma has the ability to provide elasticity to the growing organs of the plants. So, that is why, as I just said, that collenchyma allows plant organs to stretch and elongate. However, sclerenchyma occurs in areas which have stopped elongation, because it does not allow any plant organ to elongate. Then collenchymatous cells have high refractive index, so they can be visualized under the microscope very easily, but the refractive index of sclerenchyma is comparatively lower. And since the collenchymatous cells are living, they can store food and take part in photosynthesis if the chloroplast is present. However, sclerenchyma has no such function. It will only provide mechanical strength. So, this was about the earlier session, and let us now begin today's session, and in today's session we will discuss the various tissue system of the plant. So, we will have a look on the epidermal tissue system, the ground tissue system and the vascular tissue system. Beginning with the epidermal tissue system. You know that on a plant, the epidermal tissue system comprises of epidermal cells, stomata and epidermal appendages, which can be the hairs or the trichomes. Talking about the epidermis. Here you can see an image of microscopic view of a leaf peel. Similar peels you must have made in the lab in junior classes also, and must have visualized them under the light microscope. You can clearly see that the epidermal cells appear to be somewhat rectangular and there appear to be no intercellular spaces. The term epidermis has been derived from a Greek word meaning over skin. Here the cells are compactly arranged and elongated and they form a continuous layer. This is what you have just seen. Can you tell me why is it so? Why the intercellular spaces or the gaps are absent? Is there any reason? Yes. The reason is that the epidermis serves as a barrier between the external environment and the internal environment of the plant body. So, for a barrier to be effective, it should be proper, it should be continuous, so that any invader or intruder cannot gain any entry. If there is any opening on the epidermis, it is highly regulated. Can you tell me the name of that opening? Yes, that is the stomata. So, except for the stomata or the stomatal pores, the epidermis forms a continuous layer over the plant body. So, it is just like our skin. Now, the epidermis is made up of parenchymatous cells. They have small amount of cytoplasm and a large vacuole. Talking about the functions of epidermis, it serves many functions. It protects against water loss and even attacking microbes, then regulates the gas exchange, secretes metabolic compounds and also absorbs water and mineral nutrients. Coming over next is the stomata, the second most important structure of the epidermal tissue system. So, you have studied in junior classes that in dicots the stomata has guard cells which are kidney shaped, but in monocots the guard cells are dumbbell shaped. In an stomatal apparatus, what do we see? We see that there is a pore. That pore is known as stoma or plural stomata. This pore is guarded by two cells. In dicots, these cells are kidney shaped, and in monocots, these cells are dumbbell shapes. These cells are known as guard cells. Besides guard cells, there are many other cells associated with the stomatal apparatus. Generally, their number and shape is of taxonomic importance. Otherwise, also it is seen that they seem to regulate the opening and closing of stomata with the help of guard cells. These cells are known as subsidiary cells. These are the guard cells, and this is the stomata, the pore. This is a diagram of an stomatal apparatus. Now, have you studied how do the guard cells help in opening and closing of stomata? Yes, you have some idea, you have studied that in junior classes, that the guard cells have numerous chloroplasts, they carry out photosynthesis, and when photosynthesis done, a lot of starch is made up in the guard cells, and when the starch is produced, the water moves inside the cells because of osmosis. As a result, the guard cells swell up and the pore opens up. But don't you think somewhere something is lacking? Suppose I use these balloons to make up the guard cells here. Suppose this is an stomatal aperture when it is closed. These green and purple balloons serve as guard cell in our experiment here. So, the stomatal aperture here is closed. Now, if somehow water enters into the cells, we say that the stomatal aperture should open up. What will happen if I just blow air into these guard cells balloons? They should open up the aperture. So, let us see. These are the two balloons of the same size that we were using as guard cells, and now I have placed them just in the place of guard cells. What has happened? Instead of opening of the aperture, the guard cells have come even more closer, and if there was any gap, it must have been closed by now. So, that means only the ability of guard cells to carry out photosynthesis is not going to help them to open the stomata. There must be something else, and that something else is the presence of uneven thickenings. I will draw the guard cells once again for you. This is the shape of a guard cell in dicots. We see that the guard cells are very thick on one side and such thickenings are absent on the other. Some radial thickenings are also present on these guard cells. So, a guard cell should look like this with uneven thickening here and here. Now, what will happen? Because of these thickenings, when water moves in, these walls can stretch, but these walls can't stretch. As a result, these walls will be pulled outside and the stomatal aperture or the pore will open up. Let us see how this happens. So, I hope you remember our balloon guard cells, and I have used a sellotape to serve the purpose of the thickenings here. Here you can see that I have put sellotape on one side to serve the purpose of thickening. And now, let us see what happens if air is blown. Air here is representing the water that is moving inside the guard cells. Let us see what happens. Here, do you see any difference in the shape of the balloon now than the earlier one? I have kept the earlier one for you also. This was the balloon without any thickening or the guard cell without any thickening. When the water came inside, it swelled up. While on the other hand, here you can see in this guard cell, so called guard cell, because of the thickening, the swelling is not even. Now, if two such guard cells are placed side by side, Never mind. So, now what will happen if two such guard cells are placed side by side? The stomata will open. This was a closed stomata and this now is the stomata open. So, you can see that because of uneven thickening of the guard cells, the stomatal pore can open or close. This is how guard cells regulate the opening and closing of the stomata. The same is true for the monocots also, though I have taken the shape of the dicot guard cells, but in case of monocots the same happens. Now, here in this slide you can see a microscopic view of the guard cells. The stomatal aperture is quite clear and you can also see the subsidiary cells. Now, the epidermis of the plants also has hairs or trichomes. Their function is absorption, secretion and protection. These are microscopic images of trichomes and hairs. You can see this hair is unicellular while these trichomes are multicellular. Trichomes serve many purposes like absorption of water and minerals as in root hairs, secretion of various oils as in mint plant. Then their main function seems to be protection. They protect against herbivory as in nettle, and they also give protection against frost and wind, while its nose they form some umbrella like structure to keep the frost away from the surface of the leaf, and they also can trap many insects. Bean leaves have been used historically to trap bed bugs in houses in Eastern Europe. The trichomes on the bean leaves capture the insect by impaling their feet. Next, we move on to the vascular tissue system, and when we talk about the vascular tissue system, we are talking about xylem and phloem. So, xylem and phloem can be arranged variously in a plant organ. When they are arranged at different radii, then the vascular bundle is said to be radial. Here in this figure, you can see that xylem and phloem are lying at different radii in the plant organ. Such radial arrangement is present in the roots. Here in this microscopic image, you can see these are xylem and these here are phloem. Can you follow the cursor? These cavities are of xylem and while these cavities are of phloem. So, xylem and phloem both here are lying at different radii. Such a vascular bundle is known as radial vascular bundle. Conjoint vascular bundles where xylem and phloem both are joined together. These two are conjoint vascular bundles. Can you see any difference between these two? Yes, here the cambium is present and the cambium is absent here. Now, because of this presence of this cambium, which is a meristematic tissue, what will happen? When this cambium becomes active, it will produce more xylem cells towards this side and newer phloem cells towards this side. So, the growth of this vascular bundle is open. Hence, such vascular bundles are known as open bundles, while those without cambium are known as closed bundles. And yes, the open bundles are present in dicots and closed bundles are present in monocots. So, moving on, I will now show you a microscopic image of a vascular bundle. Can you identify the parts? Yes. Can you tell me what part is this? Yes, you are right, this is phloem and this is xylem. Can you identify which type of vascular bundle is it? Of course, it is a conjoint one, and since the cambium is not there, this is a closed vascular bundle. Now, we will discuss the anatomy of dicot and monocot plants. We will study the different structures in detail of organs like stems, roots and leaves. Let us begin with the dicot root. Here this diagram is given in your NCERT books also, and we will discuss the features of dicot's root one by one. Beginning with the epidermis, the cells are barrel shaped and the root hairs are unicellular. Cortex is many layers thick, is made up of parenchymatous cells and has intercellular spaces. Endodermis is the innermost layer of the cortex. Here, this layer will be known as endodermis. It is only a single layer of cells, but the cells are barrel shaped with no intercellular spaces. A special feature, that is the Casparian strips, are present here. Moving on, the next layer that comes inside is the pericycle. The pericycle is few layers thick and it has thick walled parenchymatous cells. The lateral roots and secondary roots initiate from here only. All the tissues that are present inside of pericycle comprise the stele. So, it includes the pith and the vascular bundles. The pith is inconspicuous in a dicot root and vascular bundle are radial. There are only 2 to 4 patches of xylem and phloem. Now, tissue that is present between xylem and phloem is known as conjunctive tissue. What I want to ask you guys is, do you think that it is so easy to cram up all the details of the structure, or will it be better if you are able to draw the diagrams and then explain the structure? The diagram given in the book can be a little bit difficult to understand if you do not know the proper anatomy of the cells. Once you learn how to draw various types of cells in a plant, then these diagrams will be very easy for you to draw. So, let us see how do we draw. We are going to draw the TS of a dicot root. First of all, we mark a triangular region in our notebook, because we are going to show a part enlarged. There are two ways of making a diagram. We can draw a line diagram or cellular diagram. Generally, we draw a line diagram and then give a view, a cellular view of a part enlarged. So, let us first draw a outline diagram of a dicot root. As we said, the outermost layer is the epidermis, followed by a few layers of cortex, and the epidermis bears unicellular root hairs. The cortex is continued until endodermis. Then there are few layers of pericycle. Why am I using different colors? To make the diagram more appealing visually? No, actually I am using the colors here to differentiate between different tissues. And then we will draw the xylem and the phloem. Do you remember that roots have exact condition, that is metaxylem is towards inside and protoxylem is towards outside. In this manner, we have drawn here an outline diagram of the dicot root TS. It is important that we label this structure properly. So, beginning with the root hairs, the epidermis, the cortex, then endodermis, Now, you must be thinking that this is so easy. Of course, it is very easy once you remember the names of different layers. But this outline diagram will help us to draw a part enlarged, that is the cellular diagram. So, xylem and phloem. Now, let us try to draw a part enlarged of dicot root so that we can show how different cells look like under a microscope. We begin with the epidermis. The epidermal cells are somewhat barrel shaped. Please pay attention as to how am I drawing these cells. Each type of cell has a different shape, be it the cells of epidermis, be it the cells of cortex, the endodermis, xylem or the phloem, and we need to keep in mind the different shapes of the cells while drawing these diagrams. So, I have drawn this epidermis, and now we say that there is a cortex which is few layered thick. Initially, in the earlier layers or the first few layers, the cortical cells are smaller, but as we move ahead, we see that the size of the cortical cells becomes larger. I will not draw in the whole area, I will draw just a little bit, so as to give you an idea how to draw the cells. If you draw complete circles with a steady hand, you will be able to draw proper shape of the cortical cells with proper intercellular spaces as well. So, we had studied that the cortex is a few layered thick, and the last or the innermost layer of the cortex is the endodermis, and again the shape of the cells of endodermis is also very important. They are barrel shaped cells and tightly packed. So, this is how I am going to draw the endodermis. So, these are the barrel shaped cells of the endodermis here, and we have said that Casparian strips are present on the endodermis. Casparian strips are made up of suberin. Suberin is impervious to water and it does not allow the water to move through the walls of endodermis in the Casparian strips. Following the endodermis, there are a few layers of cells which are thick walled and make up the pericycle. So, this is how I am going to make up the cells of pericycle, and after drawing them, I will give them a little bit thickening like this. This needs to be done for all the cells, but I am going to do it for a few only. Then comes the xylem and the phloem. As we said that the protoxylem is towards the outer side. We will try to draw some polygonal cavities showing the protoxylem, that is the smaller and narrower cavities, and larger cavities will be used to show the metaxylem. This is how we can draw xylem. And besides xylem, we will also draw phloem. When we draw phloem, we generally draw a sieve tube cell and a companion cell with it. This is how we draw phloem. We show nuclei in some of the cells, and the cells containing nuclei are the companion cells, and this is it. We can also draw some conjunctive tissue with a very light hand, because the spaces are not empty here, there are cells everywhere, right. So, the conjunctive tissue will also be drawn in this manner. Now, let us label. This is root hair, the epidermis, the cortex, the endodermis, few layers of pericycle, phloem, xylem and conjunctive tissue. This is how we can draw the TS of a dicot root. If you practice drawing the cells, it will become very easy for you. Look at the diagrams of monocot root and dicot root. Can you see the differences between the two? Most of the upper layers are similar. The main differences lie in the stele. That is, here we see only 2 or 4 patches of xylem and phloem, but here the patches are many. So, when we discuss the monocot root, we say that the structure of epidermis, cortex, endodermis and pericycle, all these are similar to the dicot root, but the stele is different, because here the pith is large and well developed, which was not present there in the dicot root, and the vascular bundles are polyarch, because there are 6 or more patches of xylem and phloem. The conjunctive tissue is also present here. This was about the roots. Let us now move on to the dicot and monocot stems. Yes, since it is monocot root, so there will be no secondary growth. Can you tell me whether it is monocot or dicot root? Of course, it is monocot root, because it has so many numbers of xylem and phloem. So, this is epidermis, cortex, what is this? Endodermis, followed by a few layers of pericycle, and then these patches there are of phloem, the larger ones of course xylem, and there is the pith in the middle. These cells, pericycle, phloem, xylem and pith, they constitute the stele. Moving over to the dicot stem. Just as we had discussed the structure of a dicot root beginning with the epidermis, here we will also see that all the layers mentioned in the root are present here, but the difference is that the epidermis bears trichomes, stomata and even the cuticle may be present. The cortex has 3 zones: hypodermis, which is made up of collenchymatous cells. Then there is cortex, which is similar to that of the roots, and endodermis. Now, in the endodermis, the Casparian strips are not present and they are rich in the starch grains. Pericycle is also different because it is present above phloem in form of semilunar patches, and it is made up of sclerenchyma. One new structure that we find here is the medullary rays. They are arranged between the vascular bundles. The pith is initially well developed, Another difference between root and stem is that the vascular bundles here are conjoint, open and dark, and they are arranged in a ring. This is how a dicot stem looks like. So, let us try to label it. First, there is epidermis, then collenchymatous hypodermis, followed by cortex, and then the innermost layer of the cortex is the endodermis. Pericycle is present in the form of semilunar patches over the phloem. ये dark green color present here is pericycle. Then there is pith. This light green patches are of phloem, and these are the xylem elements. And can you tell me what are these white strips present between xylem and phloem? Yes, they are the cambium, and they are present between xylem and phloem. The anatomy of a monocot stem is quite different from that of the dicot stem. Why? Because the hypodermis here is sclerenchymatous, while that there in the dicot stem it was collenchymatous. Vascular bundles are also different. They are scattered, conjoint, closed, and even the bundle sheath is present. Further, the phloem parenchyma is also absent, and the bundles may have water containing cavities in them. This is the figure from your NCERT book, and you can easily see the scattered vascular bundles and identify it as a monocot stem. This is a microscopic image of a monocot stem, and here you can see the scattered bundles. The tissue between is known as ground tissue, and the layer of cells that surrounds each bundle is known as bundle sheath cell, and these are the scattered bundles here. Talking about the dicot leaf. The leaves have different epidermis on the upper and lower side. On the upper side, the epidermis is known as adaxial epidermis. It has conspicuous cuticle. The lower epidermis is known as abaxial epidermis. It has more stomata comparatively. Then the mesophyll is of two types, palisade parenchyma and spongy parenchyma. The vascular bundle, the main vascular bundle is that of the midrib, and bundle sheath is also present. Let us see this diagram that is given in your NCERT. So, this is the upper epidermis and the lower epidermis. The upper epidermis is covered by a cuticle, and the epidermis is followed by long elongated finger like cells known as the palisade mesophills. Now, trick to draw the palisade cells is that you draw continuous U without any break, without uplifting your pen or pencil with which you are drawing. In this way you can draw the palisade cells. These dots, they show the chloroplast. Now, the spongy parenchyma is the one where the air cavities are present for the gaseous exchange, and this is microscopic image of the same dicot leaf, and you can see the upper epidermis, the palisade mesophyll, the air cavities, the spongy mesophyll and the substomatal cavity for a good gaseous exchange. The lower epidermis, that is known as abaxial epidermis, is also visible. The anatomy of monocot leaf differs from dicot because it is isobilateral, that is both the sides are similar, and the mesophyll cells are of only one type. The special feature is presence of bulliform cells. When bulliform cells absorb water, they are turgid. The leaf is well exposed, but when the water is scarce, they become flaccid and the leaf curls up to prevent the loss of water. Here these structures, these are the bulliform cells. So, here this is the diagram of a monocot leaf that is given in your book, and this is a microscopic image of the same leaf, that is a monocot leaf. Can you see how identical these features are? Well, for once the upper and lower epidermis, they are very similar. That is why the monocot leaf is known as isobilateral leaf, and you can see that the other structures are also very clear. this is the vascular bundle present here, and the water cavities and the xylem and the phloem. So, this was all about today's class, and in this class we studied in detail the epidermal tissue system, how different cells and structures of epidermis are organized and how they function. Then we studied about the anatomical details of the root, stem and leaves of dicot and monocot plants. Since we have studied so much anatomical structures, it will be very good if you try to differentiate between the following: the anatomy of dicot stem and dicot root, monocot stem and monocot root and dicot root, and monocot and dicot leaf. If you are able to differentiate between these dicot and monocot stem also, if you are able to differentiate between these, that will mean that you have clearly understood the structural organization in these structures. I have drawn some hints for you to write your answers and frame your answers around these points. Do not copy them, just use them as hints. So, well, when we differentiate between dicot stem and dicot root, we can use these points. The trichomes on the stems can be multicellular and or branched too. The stomata and cuticle may be present. The hypodermis is collenchymatous. Endodermis cells are rich in starch grain. Pericycle is present only above the phloem, and that too in semilunar patches, and also it is made up of sclerenchymatous tissue. The medullary layers are present in the dicot stems. The pith is well developed initially. Later on, when secondary growth takes place, the pith is crushed, but earlier in the earlier stages of development, it is quite well developed. Talking about the vascular bundles, they are conjoint and open, and also the protoxylem is endarc. Compare it now with the dicot root, where the root hairs are always unicellular, and the other features like the cortex and hypodermis, the hypodermis is absent. The endodermis, it is made up of Casparian strips. Pericycles, it is few layers thick and it is made up of parenchyma. Medullary rays are absent in the dicot root, but conjunctive tissue is present between the xylem and the phloem. The pith is inconspicuous and the vascular bundles are radial with exarch protoxylem. The next was to differentiate between dicot stem and monocot stem. So, here are the differences. In dicot stem, the hypodermis is collenchymatous, endodermis, pericycle, they all are present. Vascular bundles are conjoint and open. The vascular bundles here are arranged in a ring, and the bundle sheath around a vascular bundle is absent. Phloem parenchyma is well developed, it is present there. Medullary rays are also present. The pith is well developed initially and water containing cavities are absent inside the vascular bundles. They may be present elsewhere, but inside the vascular bundle they are absent. Now, let us compare these structures with the monocot stem. So, the hypodermis here is sclerenchymatous, endodermis, pericycle, these structures are absent, and vascular bundles, well, they are conjoined and closed. Also, they are not arranged in a ring, but they are scattered. The bundle sheath is present around them and phloem parenchyma is absent. Finally, medullary rays and pith are also absent and water containing cavities are present inside the vascular bundles. This is how you can differentiate between a dicot and a monocot stem. Moving up ahead, let us try to differentiate between monocot root and dicot root. Obviously, the first difference is that no secondary growth occurs in a monocot root. No secondary growth occurs in any of the monocot structures, but secondary growth occurs in the dicot root. Other points of differences are about the pith, Well, it is large and well developed in the monocot root, but it is inconspicuous in the dicot root. Vascular bundles, they are polyarc in the monocot root, but they are only in 2 to 4 patches in dicot root, since the secondary growth is not there because cambium is absent, and the secondary growth here is because of presence of cambium. Last is the difference between monocot and dicot leaf. The first prominent difference is that monocot leaf is isobilateral, while dicot leaf has adaxial and abaxial surfaces. The mesophyll cells here are only of one type, while there are two types of mesophyll cells, spongy and palisade parenchyma. Last difference is that the bulliform cells are present in the monocot leaf, but they are absent in the dicot leaf. This was all about the questions that I have asked you to frame answers for, and in the next session we will study the secondary growth in stems, vascular cambium, springwood and autumnwood, heartwood and sapwood, cork cambium, secondary growth in roots. This was all about today's session. Thank you very much.